Good morning, students. Welcome to our class Biological Science. Dear students, in the previous lesson we have already started our lesson reproduction. So in that lesson we have already discussed that what is reproduction, what are the methods of reproduction, either sexual or asexual. Especially in the asexual mode of reproduction, we observe that asexual mode of reproduction in the plants and some microorganisms. That means budding, cutting, layering, grafting, and a microorganism especially budding, formation, spore formation, fragmentation, regeneration, that type of parthenogenesis, we discussed that. Yes or no? Once you recall all the asexual mode of reproduction where a single gamete is involved, a single organism is involved and no fission of gametes, budding. Have you remembered that? Fission, budding, fragmentation, parthenogenesis, regeneration. Dear students, I am explaining once again about this. Everything is new terminology. It's entirely different with your 9th class lessons. In the 9th class, we discussed or you learned only the cell biology. The cell structure and its components, plant tissues, animal tissues, blood groups we discussed it. And something about the plants, their habitats or environment and pollution. But it is entirely different with your 9th class and new terminology is introduced here. Okay, don't confuse about the lessons. Listen carefully. When you come to school, I will explain everything to you. Eye-to-eye contact, you can easily understand about these terms are the words what I am explaining or that is present in your textbook, where they are present and what's their function. So avoid all these things, listen carefully, follow your textbooks and what I am explaining to you. Okay? So come to our lesson. So in the previous section we discussed the asexual mode of reproduction. Asexual mode of reproduction. So we discussed that reproduction in which single organism is involved and there is no fission of gametes, that is asexual mode of reproduction. So we can see this mode of reproduction in the lower animals like amoeba, bacteria, paramecium, like the fission. And second one is the budding, hydra and yeast. Yes or no? Fragmentation, parthenogenesis. So parthenogenesis, regeneration. After that, asexual mode of reproduction in the plants, vegetative propagation, naturally and artificially. Naturally, biofilm and alien victims like runners and stolons, we discussed that. Okay, artificial vegetative propagation done by our humans, that is cutting, layering, grafting. Tissue culture also we discussed. Here I am going to explain a few things about the asexual mode of reproduction. Some of the organisms or some non-flowering plants can follow a type of reproduction that is followed by the spores. I will explain about it. Spore formation. Okay, reproduction by the spores. Asexual mode of reproduction by spores. Formation of spores. What are these spores? Where they are present? Which organisms are produced these spores? I will explain about it. Generally, you can see that whitish thread-like structures and blackish powdery-like substances on a rotten slices, rotten vegetables and a bread molds, bread, eating bread. Okay, do you understand? What is that? Generally, we may see that whitish thread-like structures. They are just like this. Whitish thread-like structures and blackish powdery substances that present on the rotten vegetables or the rotten bread slices or other foods also. Okay, when you touch these materials or the substances, the blackish powdery substances sticks on our fingertips. These are the reproductive spores produced by the fungi. What are those? I already written reproductive spores produced by fungi. Example, rhizopus. Okay, now. So listen carefully, there is no confusion about it. Generally, we may notice that whitish thread-like structures and blackish powdery substances on the rotten food materials like vegetables or the bread. Okay, so when we touch these substances, the blackish powdery substances touch on or sticks on our fingertips. These are the reproductive spores produced by the fungi. Example, rhizopus. Do you understand that? Okay, in our Telugu, that is boozu. Boozu bartadi kada mana ahara padarthalu. If you keep aside all the food materials, boozu bartadi kada. So a booze to paatu black color dots form adi. Those are the blackish powdery substances, spores. Do you understand? So the spores are produced by these fungi. Rhizopus produces hundreds of microscopic reproductive units called spores. Do you understand? Rhizopus produces hundreds of microscopic reproductive units called spores. These are the reproductive units. In the previous section, we discussed that ovum and sperms together they develop into zygote. Here, a sexual mode of reproduction, the spores will develop into the new individuals. Do you understand that? The rhizopus produces hundreds of microscopic reproductive units called spores. These spores develops into individuals. The spores develops into individuals. The spores are present in the sporangium. See this. This is sporangia. Sporangium. Singular sporangium, plural sporangia. Okay, these are spores. The spores are present in the sporangium. When the spore case bursts, as sporangia bursts, the spores enters into the air. When they fall on the ground or on food or in the soil, by getting the favorable conditions like damp and dark areas, they will grow into new individual. This is the mode of reproduction. This is the mode of asexual reproduction performed by these fungi, rhizopus, okay, and mucor. Another example is mucor. Fungi, rhizopus and mucor. Okay, and some bacteria and some flowering plants, non-flowering plants like ferns and mosses. Non-flowering plants, ferns and mosses. Okay, this is the mode of reproduction that we can see in the fungi. So this mode of spore formation, we can see in the fungi like rhizopus and mucor, and some bacteria and some non-flowering plants like ferns and mosses. Do you understand this? Very simple one. Okay, whitish thread-like structures and blackish powdery substances present under rotten food materials. Those are spores. Spores are produced by these fungi. Example, rhizopus and mucor. So, rhizopus produces hundreds of microscopic reproductive units called spores. When the sporangium bursts, the spores enter into the air. When they fall on the ground or on the food or in the soil, and by getting the favorable conditions like damp and dark areas, they will go into new individuals. So, example for this type of spore formation is fungi, rhizopus and mucor, and some bacteria and non-flowering plants like ferns and mosses. Okay, so dear students, this is the mode of asexual reproduction. Now I am going to explain about an experiment. How would you observe, how would you examine the common bread mold under the microscope? Very important for my question. Your seniors get number of times in the examinations. So to examine the rhizopus or common bread mold, under the microscope. Do you understand the question? to examine the common mold under the microscope. Dear students, this is our experiment to examine, to identify. How would you identify the common mold or rhizopus under the microscope? To examine this common mold under the microscope, we need or we have to grow the mold. Okay, now we have to grow the mold in our own way. It is better to grow the mold in your own way in a controlled environment. To grow the mold, we need. That means I already told you that common mold means goose. Okay, to grow the mold, we need preservative-free roti. A roti or a bread which is preservative-free. Follow your textbooks or listen carefully. We need. So, for this experiment, we need to develop the common mold. It is better to grow the mold under the favorable conditions in your own way. To grow the mold, we need a bread slice or a roti which is preservative free. Fridge lo పెట్టినటువంటి bread ని కానీ roti కానీ తీసుకోవాలి. Okay, which is preservative free and vegetables like potato or oranges. Okay, now the common bread mold requires 4 to 10 days. Okay, the bread can grow. Okay, a mold can grow in 4 to 10 days in the favorable conditions. It requires some suitable conditions. Okay, so the bread mold will grow within 4 to 10 days in the suitable conditions. So to do this experiment, go or plan ahead. Okay, go with a plan ahead. That means we manchi avagahana undali. Do you understand here? So procedure. How do we do this one? Okay, now take the piece of bread. For example, it is a piece of bread. Take a piece of bread. Take a piece of bread and leave it in the air for about an hour. Okay, na? Leave the piece of bread in the air for about an hour. It exposes to all the contaminants that present in the air. Why do we keep the bread in the air? Because air contains all the microorganisms. Fungi, bacteria, amoeba, water. Fungi, bacteria, virus, everything is present in the air. So if we leave the bread in the air for about an hour, it is exposed to all the contaminants that present in the air. Next, seal the, place the bread in a plastic bag. Place the bread in a plastic bag by sprinkling some water over it. By sprinkling some water over it to make the dampness and seal the cover. And seal the cover by leaving some air inside. Seal the cover by leaving some air inside. That is the first step. What we did in the first step? Leave the bread in the air for about an hour, so it is exposed to all the contaminants in the air. The next step, place the bread in a plastic bag and sprinkling some water over it to make the dampness and seal the cover by leaving some air. Next, place this plastic bag or place the bread in a dark and damp place, in a dark and warm place. Dark and warm place. A kitchen cupboard close to the stove is a good place for you. Or next to the window by covering with the lid or bowl to prevent from the light. Okay, so this one is better. Place the bread close to the gas stove in the kitchen cupboard. Do you understand? Place the bag or place the bread in the kitchen cupboard next to the stove. Okay, so the bread mold start to grow within 2 to 3 days. The best mold grows. The growing of best mold takes a week, 1 week or more. You understand? Place the bag in the kitchen cupboard very close to the stove because the growing of mold requires suitable conditions, dark and damp conditions. Do you understand that? To grow the mold, it requires dark and damp conditions. The mold grow within 2 to 3 days, but it takes 1 week or more to produce the more spores. To produce the more spores, depending on the conditions, weather conditions. Do you understand? Depending on the weather conditions. That is the first paragraph or the first step we have done. In the first step, to examine the rhizospores, we have to grow the mold in our own way. To grow the mold, we need a bread or roti which is preservative free. The best mold grow within 4 to 10 days. It's depend on the climatical conditions. Okay, now in the procedure, the first one, take the bread and leave it in the air which is exposed to all the contaminants that present in the air. Next, place it in the plastic bag and sprinkle some water over it. Okay, and seal the bag by leaving some air inside. Why do we sprinkle the water? To make the dampness. Because next, keep it in the dark and damp places. Okay, so because it requires the suitable climatical conditions. The mold would start to grow within 2 to 3 days, but the more spores are the best mold will form in a week or more days depending on the suitable conditions that present around us. Next, in the second step, observe the piece of bread after every few days. Observe the piece of bread every few days and sprinkle water over it if it is drying. And sprinkle water if it is drying. Avoid the opening of plastic bag as far as possible because it causes some allergic senses. Do you understand that? So observe the mold. Observe the mold after every few days. If it is drying, add some water to it. If it is possible, avoid the opening of this bag or the bread, because it causes allergic senses. So the bread mold has totally grown. If it is grown, that mold is grown. Take that piece of it and slide and observe under the microscope. There you may notice that whitish thread-like structures, and blackish powdery substances. Whitish thread-like structures and blackish powdery substances, black, grey or green color powdery substances. The whitish thread-like structures and black dot structures that appears on the slide is the bread mold. What is that? Bread mold. Do you understand? So in this way we develop the bread mold. Okay. So this is the process of mode of reproduction in this common mold or rhizospores. Okay. In the second step, open the bag and observe the piece of the bread. If it is drying, add some water to it. In the next case, as far as possible, better to avoid the opening of this piece of bread because it causes allergic senses. Okay, next, the bread mold will grow within 1 week. After that, it forms a good mold, and take a scratch a little from it and observe on the slide. Observe on the slide. What we notice that? We notice that here. Okay, under the microscope, whitish thread-like and blackish substances which are black, grey, or green in color. These are the rhizospores. Okay, take the mold to your school in the matchbox and show to your teacher. Okay, so this is about to examine the common mold under the microscope. Dear students, in the next section we will see how to observe this common mold under the microscope. I will explain about that process. Okay, once you did all these things, thank you very much.